Hello, everyone, and welcome to another exciting episode of the Biologic Podcast. Today's show is episode 107, and it's part 2 in this two-part miniseries on the cephalopods. In the first part of this miniseries, in episode 106, I explored the early evolution of the mollusks generally, and the cephalopods specifically. By the time we got to the common ancestor of the nautilus and the coleoid lineages, we had reached a fork in the road. As the first episode turned to focus on the nautilids, we followed the nautilus lineage and explored the modern descendant nautiluses that live near the lower regions of coral reefs and continental shelves in the southeastern regions of the Pacific Ocean. Today's episode is all about the other lineage, the coleoid lineage. This group includes some of the most dynamic and amazing cephalopods: the cuttlefish, the squid, and the octopus. I can't wait to get into the physiology and ecology of these animals, but first, we need to go back to that evolutionary fork in the road to follow the coleoid lineage from that point way back in the Paleozoic when they diverged from the ancestral nautiloids, and follow them into the modern day. To put everything into context, let's do a quick summary of mollusk evolution right up until this big divergence. We started out in the Ediacaran, approximately 600 million years ago. This was a time when multicellular organisms had really started to develop increasingly large and complex forms. We saw the emergence of the Lophotrochozoans, primitive slug-like creatures with tentacles surrounding their mouth. These Lophotrochozoans prowled around on the seafloor, searching for food to gobble up with their face tentacles. About 575 million years ago, the Lophotrochozoans evolved mineralized shells to protect them from predators that swam above them. Over the next 75 million years or so, these shells increased in size and diversified in shape, with some curling forward or backward. In the Cambrian, the shells evolved internal support structures, called septa, which were then built up and organized into complete internal walls. This, in turn, created internal chambers lining the shell. This was followed by the evolution of the siphuncle, a membranous tissue that ran along the side or the middle of the shell, passing through all of these internal chambers. This siphuncle was a super important evolutionary adaptation, because it allowed the ancestral cephalopods to drain fluid out of the chambers and replace it with gas, making their shell buoyant. Some species evolved cameral deposits, or buildups of mineralized tissue inside the shell, usually near its tip, to work as a ballast or a counterweight so that the body axis could be rotated into the horizontal. And, most importantly, this allowed these animals to swim. The gas-filled shell made them buoyant, and by flapping their tentacles and pushing water out of a siphon organ, they could propel themselves forward, with the pointed hydrodynamic shape of the shell able to slice through the water. With the ability to swim, these animals became quite proficient hunters. As a clade, they produced many dominant predators in these late Cambrian and early Ordovician ecosystems. So now we're in a time period about 500 to 450 million years ago. This is still a bit early in evolutionary history, but some paleontologists think that this was when the ancestral nautiloids and the ancestral coleoids diverged. In the previous episode, I mentioned that around this time, the Elasmerocerida had produced descendant lineages in the Oncocerids and the Orthocerids. Some paleontologists think that the orthocerids represent the last common ancestor between nautilids and coleoids, but recent evidence suggests that the common ancestor came earlier, perhaps from the Elasmerocerida themselves, or even from their proximate ancestors, the Plectronoceras. The main takeaway from all of this is it's strongly suspected that the Oncocerids are the ancestors of the nautiluses, and the orthocerids are the ancestors of the coleoids, but we're less certain about when their actual common ancestor lived. We think that it was in the mid-Cambrian to early Ordovician period, but it may be even older. I'm not a paleontologist, but it's my understanding that Elasmerocerida is a popular contender for the common ancestor of orthocerids and oncocerids, and thus nautiloids and coleoids. Alright, so the orthocerids proliferated around 480 to 485 million years ago, and they would eventually give rise to the coleoids, but it would take another 150 million years for the earliest true coleoids to appear. You had 150 million years of stem groups evolving, diversifying, dying out, finding their own niches, so on and so forth. For example, there was the Bactritida, the immediate descendants of the orthocerids, which emerged around 407 million years ago in the early Devonian period. Both the orthocerids and the Bactritida had a vertical orientation. Their conical shells pointed up, and their tentacles and mouth grazed along the seafloor, searching for food. This posture is relatively inefficient for swimming, so they would not have been good at chasing down prey. Instead, they were likely ambush predators, or slow-motion hunters who stalked their prey by hiding among seaweeds and coral until finding an opportune moment to strike. These Bactritida are really interesting because they gave rise not just to the coleoids, but also to two other major cephalopod lineages, the ammonoids and the belemnoids. Now, the ammonoids are more closely related to the coleoids than they are to the nautiloids, but morphologically speaking, they were actually generally more similar in shape to the nautiloids, with their curling shells, than they were to the straight-shelled coleoids. Now, the other group, the belemnoids as a lineage, are slightly older than the ammonoids, and they're much more similar to the squid than they are to any other cephalopod. Now, these two clades, the ammonoids and the belemnoids, prowled the seafloors and the continental shelves, but they would not survive into the modern day. After the ammonites emerged from the ancestral Bactritida in the early Permian period, about 285 to 299 million years ago, they quickly became a very influential component of their marine ecosystems. Unfortunately for them, their dominance was short-lived, as they would be outcompeted by their relatives and dwindle in number and diversity throughout the rest of the Permian. This put them in a very vulnerable position for what was to come. About 252 million years ago, a huge volcanic region called the Siberian Traps began to erupt. This volcanic activity was sustained for centuries, if not millennia, across a massive land area, and the effect on the atmosphere was nothing less than devastating. These enormous Siberian eruptions belched colossal amounts of CO2 into the air, which greatly warmed the atmosphere. A lot of this CO2 dissolved into the ocean, making the ocean substantially more acidic. The atmospheric CO2 trapped heat, and this led to a rapid warming of the air, which in turn led to a rapid warming of the land and the sea. In the sea, the warmer temperatures dropped the oxygen concentration to disastrous anoxic levels. In this environmental destruction, 70% of terrestrial vertebrate species were wiped out. More than 80% of marine species were wiped out. The trilobites were destroyed completely. An ancient cousin of the sharks, the acanthodians, were destroyed completely. The mollusk clade suffered immensely, but narrowly avoided complete destruction. The bivalves were savaged, with about 59% of known genera dying out. The ammonite and the gastropod clades were both pushed near the point of extinction, with 97 and 98% of their genera destroyed, respectively. It is an understatement to say that the Earth's ecologies were utterly destroyed and remade over the next 10 to 30 million years. Only two ammonoid clades survived this Permian-Triassic extinction, the Ceratidida and the Ammonitida. Both of these groups emerged shortly before the Permian-Triassic extinction, and somehow they managed to survive it. Both groups would proliferate over the next 50 million years or so, but near the end of the Triassic, their diversity would plummet, and the end Triassic extinction event would completely wipe out the Ceratidida. The Ammonitida were massively damaged in this event too, but there was a single Amanitida family that was able to survive, a single one. This was the Siloceratidae family, which clung to existence by the tip of their beaks until diversifying wildly in the early Jurassic period, re-establishing the ammonites as a diverse and thriving clade. New clades emerged, including the true ammonites in the Jurassic, and new forms appeared, including those with smaller shells, shells with more complex ridges and folds, more complex protrusions and suture lines, as well as more tightly coiled shells, and even new filter feeding strategies. The ammonoids endured the rigors of the Mesozoic era, despite the heavy predation pressure. Let's not forget the belemnoids. These were still swimming around too, and they also enjoyed relatively high abundance and diversity in the Jurassic and the Cretaceous periods. And on the flip side, they also faced heavy predation pressure. The belemnoids had evolved a pointed spearhead shape at the tip of their shells, increasing their hydrodynamic efficiency akin to their cousins, the squid. The belemnoids also evolved hooks on their arms, allowing them to ensnare and better hold onto prey. They evolved to inhabit a broad range of marine ecosystems, from the dark sea floor and coral reefs to well-lit and heavily vegetated coastal habitats, to the dangerous open ocean itself. In the end, ultimately, the predation pressure became so strong that by the middle of the Cretaceous, the ammonoids and belemnoids went into steep decline. Species after species went extinct, and by the very final stretch of the Cretaceous period, there were fewer than 10 ammonite species left. The belemnoids were a bit better off, but not by much. 66 million years ago, an enormous asteroid hit the planet, throwing up huge amounts of dust and debris and sending a surge of heat into the atmosphere, the oceans, and the crust itself. The devastation famously destroyed the non-avian dinosaurs, allowing the mammals to evolve and diversify and fill all of the emptied ecological niches. Less well known is the fact that the ammonites were also destroyed by the after effects of the asteroid. Paleontologists have theorized that the ammonites were particularly vulnerable due to their reproductive strategy. Near the end of their life, adults will lay a single, enormous clutch of tiny eggs. These eggs, and the juvenile ammonites that hatch from them, live in the uppermost layers of the water column, where they're part of the plankton community. This means that the ammonites had their eggs and larvae near the surface of the water, near the interface of sea and sky. When the sky was superheated and filled with CO2, which then warmed and acidified the sea, this surface water environment was the most profoundly affected. It experienced the greatest changes. The nautiloids, in contrast, laid multiple smaller clutches of eggs in much deeper water. This protected them from the worst effects of the asteroid impact, and as a result, the nautiloids fared much better and they would survive to propagate into the modern day. Curiously, in the millions of years after the ammonoids went extinct, there would be many nautiloid lineages that would adapt to more ammonoid-like lifestyles, even evolving shell morphologies that previously were only seen in ammonoids. This possibly shows adaptation to shallower habitats. Alright, so we started that story about the ammonoids and belemnoids with the ancestral Bactritida, an ancient clade that emerged more than 400 million years ago. These Bactritida also gave rise to the coleoids, which are still around today. For the rest of the episode, we're going to look at coleoid evolution, which will introduce us to the modern coleoid descendants, the octopus, the squid, and the cuttlefish. It's not known for certain when the first coleoids appeared. Paleontologists have argued for years about where in the Devonian period they first emerged, with some arguing that fossils at the very beginning of the Devonian are the first true coleoids, while others argue that the first true coleoids appeared sometime later, at some other point, somewhere else within the period's 60 million year long time span. What we do know is that the first truly unambiguous coleoids in the fossil record started to show up around 330 million years ago, right in the heart of the Carboniferous. There's debate that the coleoids may actually have originated way earlier, like the late Silurian or even in the Cambrian, but all the proposed fossil specimens are kind of ambiguous, and the classification has been the cause of arguments among paleontologists. I'm inclined to think that the late Silurian estimate is the most likely, which would mean that the first true coleoids appeared around 415 to 420 million years ago, and their sparse fossil record means that we don't really get any unambiguous specimens for about 90 million years. Now, one of the most characteristic features of the coleoids was their shell, specifically the fact that they had mostly evolved to lose it, or at least to internalize it, and reduce the mass and nutrient demand of the tissue by a significant margin. So where the ammonoids and the nautiloids have big, hard, curling external shells, the coleoids had straight conical shells that gradually evolved to be internalized and then reduced, and in many cases dispensed with entirely. They just completely lost their shell. For example, in the squid, the internalized shell has been reduced to a thin rod or leaf-shaped piece of chitinous tissue called a pen or a gladius, which is used as a kind of pseudo-vertebral column. It provides points of attachment for muscles. The reason why this internalization happened is extremely fascinating. Back in episode 106, I mentioned the differences in hydrodynamics between the nautiloid and the coleoid shells. The nautiloids, with their big, heavy, curling shells, had greater balance and stability, but they were much less hydrodynamic. They were slow. This limitation made the nautiloids struggle to compete against other cephalopods and avoid predatory fish. So the nautiloids were pushed deeper to less populated waters, where competition and predation was less intense, and their big, heavy, curling shell that made them such slow, inefficient swimmers was not as much of a liability. This adaptation made the coleoids significantly faster than their more primitive kin, but it came at the cost of their protection, their armor. The early Mesozoic oceans were a dangerous place, and armor is useful. So why did the coleoids abandon their armor to prioritize speed? We will unravel the answer to this question in a few minutes. The fish had diversified wildly in the Devonian. They dominated the ocean ecosystem so much that the Devonian is often called the Age of Fishes. Among these early Devonian fish, there were a lineage of ray-finned fish, the Actinopterygii. This lineage of ray-finned fish would become incredibly successful. They enjoyed an initial pulse of diversification in the Carboniferous and a second pulse of diversification in the Cretaceous. And today, the descendants of those original Actinopterygii make up approximately 99% of all modern fish. They make up approximately half of all vertebrate species. Naturally, these ray-finned fish suffered like everything else during the major extinction events, even getting some of their major clades, some of their biggest and most diverse clades, wiped out. But overall, they were, and they continue to be, very successful. The ray-finned fish have diversified to adapt to virtually every marine habitat, and to fill virtually every ecological niche available to macroscopic fauna. Now, if you're a coleoid, this is a problem. Those fish are way more hydrodynamic than you. They're more social, they hang out in big groups, they can outswim you, they can outmaneuver you, they can beat you to the prey, and they can force you away from a kill. And if they want to eat you, there's not a whole lot that you can do about it. In this evolutionary context, only the fastest, most agile, most inconspicuous coleoids will survive, as these are the only ones that can reliably keep up with, or keep away from, the dangerously capable ray-finned fish. Okay, let's pause here for a moment. You might have noticed something. A few clues pointing towards some mysterious force that acted on cephalopod evolution. In the last episode, I mentioned a mysterious evolutionary force that affected the nautiloids and all of the cephalopods in the Mesozoic era. Earlier in this episode, I mentioned the predation pressure that ammonoids and belemnoids faced during the Middle and later Mesozoic, and I also discussed the coleoids evolving to lose their shells to become faster and more agile, due in part to the competitive and predation pressure that they faced from the ray-finned fish. The question that you should have right now is, why are the cephalopods facing such intense selection pressure from predatory fish, to the point that every cephalopod clade shows declines in abundance and diversity? The answer is the Mesozoic marine revolution, which was an evolutionary arms race that took place across much of the Middle to late Mesozoic era. It began possibly as far back as the late Triassic, around 210 to 220 million years ago, but it really picked up speed and became more evident across the late Jurassic and Cretaceous periods. Essentially, there were a lot of marine reptiles, ray-finned fish, and various benthic organisms that frequently came across the shelled cephalopods, to the point that they eventually began to evolve an ability called durophagy. This is just a fancy scientific word for shell crushing. After all, if you're a carnivore, and there's abundant food all around you, but it's encased in a hard protective shell, any mutation that makes you a more effective shell crusher, or shell breaker, or gives you some trait that allows you to drill through a shell, means you're going to have easier access to the proteins and calories of an abundant food source. When this caught on, many benthic invertebrate species with shells, including many cephalopod lineages, were quickly destroyed before they could adapt. Large marine vertebrates like plesiosaurs, ichthyosaurs, and tychodontid sharks had the jaw strength to utterly crush their shells, to crush their entire body, and smaller organisms evolved ways to crack, scrape, or drill through a shell. Many suspension feeders, which had shelled bodies, or which used corals as a substrate, were suddenly extremely vulnerable, and they suffered for it, with many of their clades dying out. There were two general responses to this increased predation risk. Some animals evolved to live deeper in the mud and the muck of the seafloor, hidden from predators. Others evolved to become more mobile, so that they could quickly move away or dodge or somehow escape their predator by outmaneuvering them. You can see that these are like two opposite reactions, two mutually exclusive strategies to deal with a new threat. Often you would see slower-moving creatures, like sea slugs, use the bury into the seafloor strategy, and you would see more mobile creatures, like the squid, go for the high-speed strategy. These shell-crushing predators of the Mesozoic marine revolution are likely responsible for the collapse of the ammonoids and the belemnoids before they were finished off by the same asteroid that killed the non-avian dinosaurs. They're likely responsible, at least in part, for keeping the nautiloids confined to deeper water in less populated habitats. And they're likely responsible for the coleoids just outright abandoning the concept of a protective shell altogether and undergoing an evolutionary shift towards speed and agility. Around 250 to 275 million years ago, there was a major divergence within the coleoid clade. This divergence produced the octobrachia and the decabrachia. The octobrachia would diverge gradually over time to produce the octopus and their relatives. The decabrachia would diverge slowly until late in the Cretaceous, when a sudden explosion in diversification produced the cuttlefish and many of the species of squid that we see today. Throughout their evolution, the cephalopods have had 10 arms. In the modern squid, 2 of these arms have been heavily altered, turning them into true tentacles. These are much longer and narrower than any of the other arms, and they end in a club-shaped pad that's covered in suckers. These tentacles can be shot out at a prey animal to catch it and reel it in towards the arms and mouth. It's not entirely dissimilar from a frog using its tongue to catch a bug. In the octobrachia, they evolved to lose a pair of arms. They didn't evolve these cool tentacle club appendages, they just lost the pair of arms entirely, leaving them with 8. this is a good time to transition from coleoid evolution to the actual descriptions of the coleoids as they exist today. There's only one major feature that truly unifies all of the modern coleoids. They all have suckers on their arms. These suckers, or suction cups, help them latch onto prey and secure them in an inescapable grip. Other features are common enough to be generally characteristic of the whole clade, but they have some variations or exceptions here and there. Two examples of such characteristics include the ink sac and the ability to alter their skin color. The ink sac is a truly fantastic trait. It's an organ that contains a melanin-rich fluid, which can be ejected from the siphon to create a dark, murky cloud in the water. This can distract a predator or even scare it off. The predator might be surprised or scared at what this big black cloud is, and this will allow the coleoid to escape. While all living coleoids possess ink, the chemical composition differs across genera and species. In the octopus and the squid, for example, the ink is generally made of molecules of melanin pigment suspended in a solution, which disperses quickly to create a sprawling, opaque cloud. However, in some species, like those that live in deeper, darker waters, they don't have ink sacs. They don't really need them, because they already live in near or total darkness. Of the few deep water species that do have ink sacs, they usually don't release a cloud of dark, obscuring ink, but rather they release bioluminescent ink that can blind, stun, and disorient attackers. The vampire squid, for example, doesn't have dark ink, but it does eject weird bioluminescent mucus from the tips of its arms. In the cuttlefish, the ink is a bit more chemically complex. It has melanin pigments, but it also has many small proteins, many amino acids, and ammonium salts that confer various defensive properties. For example, the alkalinity of the ink, or some functional amino acid group, can disrupt the chemosensors of predators, making it difficult for them to track the cuttlefish by smelling them. For example, the enzyme tyrosinase is capable of suppressing or deactivating the chemosensory system of the moray eel. In effect, this prevents the moray eel from smelling the cuttlefish, and that makes it a lot harder to track it and follow after it and attack it and eat it. If it's having trouble doing that, then the cuttlefish can make its getaway. The other trait that I used as an example was the ability to change the color of their skin. Many coleoids use cells called chromatophores to alter the coloration of their skin at will, and this confers huge advantages for these coleoids. An important part of their defensive strategy involves camouflage, where they can alter the color of their skin to blend into the background. This ability is remarkably accurate and fast-acting, with the animals able to recreate complex patterns, and in the case of the cuttlefish and some octopus, they can even recreate textures. They can change not just the color, but the texture of their skin to blend in to, say, a coral reef or rocks on the seafloor. Camouflage is generally the first go-to defensive strategy to avoid predators, but this ability is so much more powerful and useful than just hiding. By rapidly changing colors back and forth, cephalopods can actually stun fish, kind of like hitting them with a strobe light and making them easy to catch. By creating aggressive or flashing color patterns, cephalopods can also try to stun or scare off larger predatory fish. There's even a slow-motion hypnosis type of coloration seen in cuttlefish, where the cuttlefish will send slow waves of dark coloration down the length of its body, kind of like the shadows of clouds rolling over a landscape. These dark bands of color are separated by lighter colors, and this slow-moving, mesmerizing pattern, drifting seamlessly over the cuttlefish body, can supposedly hypnotize prey animals, making them dazed and slow to react, and thus easy to capture. The active skin coloring can also be used for communication and mating displays to other members of their species. For example, some species might use one particular set of specific displays to attract mates, and then they might use a different set of specific displays to scare off competing suitors. Besides the suction cups on the arms, the ink, and the camouflage, every other trait and feature that generally applies to the coleoids demonstrates great variation between species, and some species may not even have that trait or feature at all. For example, consider the size of the eggs and the juvenile life stage. In many octopus species, for example, the eggs are generally small, and they hatch into free-swimming paralarvae, which look like weird, underdeveloped embryonic versions of their adult form. These octopus larvae are virtually microscopic, and they exist as part of the plankton until they grow larger and mature. But some octopus species, and all of the squid, lay relatively large eggs. In these larger eggs, the offspring skips the larval stage. It grows through the larval stage in the egg, and it isn't part of the plankton for long, or at all, and it hatches as a fully formed, but just smaller version of the adult body shape, instead of hatching as a weirdly underdeveloped embryonic-looking thing. Once they're born, the coleoids can look forward to a painfully short lifespan. Many smaller species of squid and octopus live no longer than a year, while larger species may live for just 3 to 5 years. Even the legendary species of enormous squid are short-lived. The giant squid is believed to have a 5-year lifespan, and the colossal squid is believed to have a mere 2 to 3 year lifespan. The craziest part of this, to me, is that this means that these squid, they have to grow from a few millimeters after being hatched to 10, 11, 12, or even 13 meters long as an adult, in only 2 years. To put this in perspective, it would be like an average full-term baby being born at about 20 inches tall, which is pretty normal, and then growing to be like 3,000 or 4,000 feet tall by the time they're 2 years old. It's just an absurd growth rate. Regarding octopus, there's been experimental evidence demonstrating that as the octopus ages, its optic glands will secrete hormones that alter the function of other organs. These optic glands, as the name suggests, are positioned near the eyes, and the hormones they secrete can induce maturation of the sex organs, which eventually allows the individual to mate and reproduce. But at the same time, the optic gland hormones also shut down the salivary and digestive glands. So after a certain age, the digestive system shuts down, and the octopus starts to starve. This period of shutdown and starvation, which will ultimately lead to the octopus's death, is known as senescence. In males, senescence takes them down relatively quickly after mating. There are some species that can mate more frequently throughout their life, but in most species, their whole lifespan is just a build-up to a big mating event, and then they die. It's generally the same for females, but just a little slower. The process is a little more prolonged. The females will lay the eggs and brood them for a short time, only to die of starvation and exhaustion shortly after the eggs hatch. In both males and females, senescence is characterized by a loss of appetite, a lack of feeding activity, and subsequent weight loss, as well as more random, undirected, or uncoordinated movements, and the appearance of white lesions on the skin. It's been debated whether these lesions are part of the senescence process itself, or whether they're symptomatic of an opportunistic infection, which is made possible due to senescence suppressing the octopus's immune system and reducing its healing abilities. Females brooding their eggs are in a senescent state, where their behavior is much more lethargic, unresponsive, and passive. It's been argued that this hormonally timed maturation, which creates a rapid life-death cycle, is actually an evolved means of avoiding overpopulation. Theoretically, if the optic glands could be removed, and their hormonal influence on the reproductive and digestive organs also removed, the octopus could live longer. An experiment in the 70s found that this is actually what happens. Removing the optic glands and their hormonal influence on other organs leads to larger body sizes and longer lifespans. So these squid and octopus can totally live longer. It's definitely within the realm of possibility. Their bodies can do it. It's just that their hormone system leads to a terminal breeding event, followed by their induced death. And the whole idea is to avoid overpopulation, because they can produce relatively large clutches of eggs, so they can grow their populations really quickly. But if their lifespan is really short and the adults die right after breeding, it's an evolved way to control their population sizes. Anyways, let's look at what the coleoids are doing during their brief life, when they're not having death sex and falling into terminal cephalopod disintegration. During their life, pretty much all of the coleoids are predators that eat a diet of crabs, shrimp, worms, bivalves, and fish of various sizes. The octopus generally trawl around on the seafloor, using their arms to turn over rocks and feel around in the sand and on coral and rock looking for prey. Cuttlefish are ambush predators, and squid generally use a more direct, chasing after you hunting style. It's more forceful and aggressive. When it comes to the squid, small, soft prey is typically ripped apart with tentacles, arms, and beak. Smaller, hard-shell prey has to be drilled into or scraped or crushed until the shell can be penetrated. Larger prey animals have to be tackled or grappled. The suckers allow the cephalopod arms to stick to the prey animal, and the constricting arms will pin down fins and prevent the prey animal from fighting back or swimming away. In some deep water species, bioluminescent effects are used at this point to stun or further disorient the animal in the chaos of the attack. In addition, the cephalopods often use toxins to help subdue and capture prey. Some species have a paralytic toxin in their saliva, which they can inject into the prey animal to slow, numb, or paralyze them. And some of these species are so toxic that they're extremely deadly. The blue-ringed octopi of the Haploclina genus, for example, are notoriously deadly. They produce tetrodotoxins, which are a type of neurotoxin that blocks sodium channels on nerve cells. Just a few micrograms of these tetrodotoxins can induce paralysis and lead to death through respiratory failure or heart failure. Once prey has been ingested, it's broken down and digested and absorbed really quickly. Nutrients are absorbed into the blue-green blood, which is then pumped through the closed circulatory system by 3 hearts. There was a lot of weird stuff in that sentence, so let's break it down. Unlike vertebrates, whose blood is usually red due to the iron-containing hemoglobin, the cephalopods have blue-green blood because they use copper-containing hemocyanin. It's a similar enzyme, but it's slightly less efficient at moving oxygen, so 3 hearts are required to achieve a high rate of blood flow to make up for the lower oxygen carrying capacity of the hemocyanin. Okay, let's shift gears and take a quick look at coleoid senses and nervous systems. If you recall from earlier episodes in this playlist, most mollusks have terrible vision. The gastropods have low-resolution vision. The bivalves have eyes, but they're not very good either. They're mostly used for sensing the changes in light caused by animals swimming above them, or by changes in the day-night cycle. The gastropods are also all deaf, and they rely mostly, or almost exclusively, on mechanoreceptors to sense the pressure waves, and chemoreceptors to sense the odors and chemical residues of nearby animals. The nautiluses, while having bigger eyes than the gastropods and the bivalves, also have generally poor eyesight, because they don't have a lens. Their eyeballs use a simple pinhole mechanism to focus light. And this is relatively primitive, and it can't produce high-quality images. In contrast to all of these other mollusks, the coleoids have extremely well-developed senses and nervous systems. Out of all the cephalopods, out of all the mollusks, out of all the invertebrates, the coleoids have the best vision and the most powerful brains. In fact, it's not inaccurate to say that coleoids have better vision and greater intelligence than most vertebrate species. This makes sense, too, as light plays a huge role in their lifestyle. To communicate using their skin, they need to be able to see the color patterns and the behaviors of other individuals. To avoid predators, they need to be able to see them from a distance to have enough time to escape or engage in camouflage or use their ink. Deep-sea coleoids, which live in the darkness for most of their life, have evolved huge eyeballs. The colossal squid has the largest eyeballs of any animal on the planet. They're approximately the size of footballs. Here's the craziest part. Most cephalopods, including all of the octopi, are colorblind. Where humans have different photoreceptors that can respond to different wavelengths and thus let us see color, the cephalopods have only one type of photoreceptor. They can't differentiate between colors. So then that begs the question, how on earth do they create such accurate color-coded camouflage? It's actually not fully understood how this works, but it's suspected that some subtypes of chromatophore don't actually produce color, but instead reflect the light from the environment. So it's more of a passive thing, like a mirror reflecting light, than any intentional color calibration. The cephalopod just has to activate these particular types of chromatophores, but they don't necessarily deliberately choose what color it's going to produce. However, recent research has identified a mechanism by which coleoids might be able to detect color. They exploit a phenomenon called chromatic aberration. Now, I'm not a physicist, and I can't claim to understand this beyond the most basic fundamentals. Hell, I can't really claim to understand that at all, but I think it works like this. So light has a wavelength, and depending on the wavelength, the refraction index is different when that light passes through a transparent object, like the lens of an eyeball. What this means is that if the lens tries to focus some incoming light, the various colors in that light, the various wavelengths, will have different refractive indices, so the lens won't focus them on the same spot of the retina. The effect is that the image is slightly distorted, and the borders between light and dark parts of the visual field can be blurred. Somehow, amazingly, the coleoids are able to interpret this blurring and distortion, this chromatic aberration, into useful information about the colors in their environment. Yeah, it's nuts. I don't understand it, but it's been experimentally demonstrated. Now, if you're smarter than me and you understood that and you thought that was mind-blowing, check this out. Let's wrap up this episode by talking about coleoid intelligence. And make no mistake, these creatures are freakishly intelligent. It's been theorized that their intelligence stems from the fact that they are active hunters. They're predators who have to seek out prey by problem-solving and strategizing. Many of them, but not all, are also very social, which is believed to contribute to the evolution of greater intelligence, as this involves navigating the nuances of social relationships, of communication and mating cues. Cephalopod intelligence is quite alien to us, and it's difficult to describe or conceptualize. But the objective cognitive abilities they've demonstrated in laboratory conditions and in the wild are undeniable. First, let's look at the anatomy of the brain and the nervous system. It's super weird. Where our brain is a single globular mass, the coleoid brain takes the form of a ring. Weirder still, this ring wraps around the esophagus. The esophagus literally goes through the brain, like sticking your finger into the hole of a donut. This strange design has a critical flaw. If the cephalopod swallows a chunk of food that's a little too big, the esophagus is going to bulge out when the bolus passes through. This bulging caused by the too large of a bite moving through the esophagus can damage the brain. It can even rip the ring apart, which is instantly fatal. Beyond the brain ring, there are large ganglia involved in other processes. A pair of ganglia above the esophagus is connected to the eyes and to the skin, where sensory information can be received and rerouted to the main brain. Below the esophagus, another pair of ganglia controls motor activity of the rest of the body, including the arms, the beak, the tentacles, etc. The arms themselves are innervated with unusually thick nerves. The cephalopods don't have myelination. They never evolved to myelate their nerves, so they make up for it by increasing the cross-sectional area of the nerves. They grow thick nerves. The end result is stuff like the giant squid axon, which has made studying neural cells really easy because it's so enormous and it's not layered in myelin sheathing, so we can directly look at the phospholipid membrane and see all of the various proteins embedded within it. Despite the brain and the nervous system taking on a relatively strange, ring-shaped form, the coleoids are, like I said, startlingly intelligent. They have very advanced spatial learning, which means they can remember pathways through a landscape, or figure out pathways through a landscape just by looking at it and thinking about its options. They can plan for the future. They exhibit enough self-restraint to enjoy delayed gratification, which is an ability that many human children don't develop for years. Communication is also very important. Consider that they can communicate with not just their body language and their movement, but also with their skin coloration and their texture. This means they have very complex communication systems with multiple interacting variables. Decoding these complex social cues, especially when you're looking at quickly changing skin coloration and skin textures, it's not easy. They also have a surprisingly sophisticated ability to problem solve. They can solve puzzles to get food. They can manipulate objects in a multitude of complex ways, due in part to the flexibility of their arms. And they not only enjoy, but actually benefit from playing around, from playing with toys or other animals. There's evidence to suggest that coleoids have a high degree of neuronal plasticity, meaning they can get bored, they can learn, they can develop preferences and habits, and they show more activity and better health when they're mentally stimulated with, quote, enriching environments, unquote, that have lots of stimulating things to interact with. There have been octopus observed using tools, and not just one tool, but a variety of different tools with different functions. Some species will also engage in construction, not the passive biological construction of growing a shell or something, but the literal act of taking building materials like bone or coconut shells or something, manipulating them or altering them, and then integrating them into a larger structure. They build walls to block the opening to their den. It's also believed that coleoids have developed such high intelligence in part due to the complexity of their prey. Crabs, for example, can fight back. They have dangerous pincers and hard shells, and it requires a pretty determined and clever foe to hunt and kill one without getting too badly hurt in the process. On this note, some species have been observed using weapons. The common blanket octopus, Tremoctopus violaceus, has been seen holding the severed tentacles of the extremely dangerous Portuguese man-o'-war. It uses these highly toxic severed tentacles as a whip, like a threatening weapon to discourage predators and to hurt or kill prey animals. That is absolutely wicked. And with that, I think we can wrap it up. I think we've come to the end of the episode. This was one of my longer episodes. And that's alright, because the coleoids are just so freaking badass that they deserve a big, in-depth episode. Now that we've wrapped up this mini-series on the cephalopods, we have one more episode in this series on the mollusks. In episode 108, we'll be looking at the relationship that mollusks have with humans. You might remember that I did an episode like this at the end of the Kingdom Plantae series, and the Kingdom Fungi series. I'm going to be doing a series on each of the major animal clades, and each of these series will end with an episode on how humans interact with the animal clade in question. So if that sounds interesting to you, if you want to learn about how humans have perceived mollusks, how we've integrated mollusks into our myths and legends and cultures, how we've used them for food, for art, for industry, and for countless other things, then hit that subscribe button so you can listen to the next episode right when I post it. Hit that like button, go to our Patreon page and become a Patreon supporter. Tell your friends about the podcast. And as always, thank you so much for listening.